Okay, in this session, what I would like to share with you is a complementary slides where will help you to understand when you do the determining of sex part one. And what am I trying to show here is there are three types of gene structures: the WT1 as an example, and then SF1 as an example, and finally the SRY gene. Now, WT1, I would say that is the normal gene. There are so many genes. If the numbers are right, there are about 30,000 genes in human, and majority of them will have this kind of structure. Whereas very handful of them will have the SF1 profile, and very few will have the SRY type of gene structures. So I'm just going to show you as an example for this variance in the gene structure, using the central dogma as a model. Now you've known this is the molecular biology for WT1. Now WT1 is found on chromosome 11, short arm of chromosome 11 at position P13. Okay, the size, as it says, is 345 kilobase. Okay, 345 kilobase. It has 10 exons. It has 10 exons. The sizes can be different range. That's why I have intentionally put the size of the exons as different sizes. Okay, they will not be uniform. Okay, now with the help of transcription process, where the enzyme RNA polymerase acts on the gene, a pre-mRNA is made. The pre-mRNA is usually the same size as the gene itself. Okay, now in eukaryotic central dogma, you have a very specialized process where we call the spliceosome will act to remove the introns, okay, during RNA splicing. Now the introns are removed, then you will get the mRNA transcript. Okay, the mRNA transcript de depends on the size of the exons. Okay, the size of the exons put together. Okay, and this will be the mRNA transcript. From this mRNA transcript, then during the translation process, ribosome will translate the mRNA transcript into amino acid, which is the 429 amino acid. This is a very typical gene structure for most of the genes in the human genome. Now the next one is SF1, which is a very important gene in the sexual development in human. Now the locus is again chromosome 9, long arm 33.3. Okay, where is that? That is here, chromosome 9, long arm. Okay, it's what do you think? Is it a metaphase? Sorry, metacentric or submetacentric type of chromosome? Okay, now from that location, this is the size of the actual gene, okay, SF1, and we know that it has got 7 exons, 2, 3, 4, 5, 6, 7 exons. Now once it gets transcribed, this is the pre-mRNA, okay. Now the pre-mRNA, the introns are removed and you get 7 exons stitched together. Now what is unique about this particular gene is that the first exon will not be involved in the translation. The sequence will not determine the amino acid found in the protein. Okay, so the first exon is a non-coding exon. Okay, that is what unique about this particular gene. Okay, and this is the molecular biology for central dogma of SF1. Okay, the last one, SRY gene, as we know, it is located in the Y chromosome, short arm of Y chromosome. Okay, short arm of Y chromosome, so it should be somewhere here. Okay, SRY gene. Okay, it's found in 11.3. and that's why it is a SRY gene. Now, it's a very small gene, 3.8 kb, and the mRNA transcript, since it does not have any introns, there's no splicing required. So it's transcribed and you get the primary transcript, and the entire exon is used to produce the translation. There are only a handful of genes which has this type of structure. Okay, so that concludes. So with that knowledge, I hope that you will actually find the central dogma for SOX9 and DAX1. I've given you the molecular biology for this gene in the slides. So please go and attempt to create the central dogma for these two genes. Thank you.